Valentin, are you ready? Can we start?
Okay.
Yes, so let me start. So welcome at today's seminar. Today's speaker is Valentin Rineau, who is our colleague, member of CTS. He's working on a ground project or macroecological ground project led by David, focusing on biodiversity dynamics over the large time scales. And this will be also the today topic by Valentin, who will be focusing on the situation in oceans. So we are looking forward to it, and the floor is yours, Valentin.
Okay, thank you. So I will add only one thing, that I'm a paleontologist, so I will focus on oceans, but at a deep time scale. So I'm working with David and his team on the project, the equilibrium theory of biodiversity dynamics. And my aim here is to present you some results, some investigations of the biodiversity dynamics and the carrying capacity through geological times. I will start by explaining perhaps what is the Phanerozoïc and why did I choose this period of time. So I'm going to give you some benchmarks of the history of life. This clock represents the Earth timeline. You can see that it begins with the formation of Earth 4.6 billion years ago. And it's mainly divided into 4 periods: the Hadean, Archean, Proterozoic, and Phanerozoic. So some benchmarks to have a look at the situation. During the Hadean, 4.5 billion years ago, we have, for example, the formation of the moon. Here it's an illustration of the Hadean. There is no record of life. Then, so it's difficult to study biodiversity dynamics without life. At the Archean, with a small jump of 1,000 million years later, we are starting to discover the first macrofossils, which are stromatolites. So here it's an example of stromatolites, where perhaps you can see the small sheets here, who are built by cyanobacteria. So it's these are sheets of cyanobacteria who grow and make a structure that are pillow-like. Stromatolites still exist today, for example, in Australia, here in shallow waters. And then let's make another small jump of 3,000 million years. So again, another thing that is important, it's during this time, we had a global oxidization of the Earth due to bacterial activity. We had the beginning of oxygen in the atmosphere, and due to oxidization in the ocean, we have precipitation of red iron beds like here. Okay, so this is a view of this period of time. You can see stromatolites, blue sky due to oxygen, and the moon, which is approximately 50,000 kilometers near from Earth than today. Okay, so the small jump of 3,000 million years. We are now during the Ediacara, so it's the end of the Proterozoic period, and we are starting to have an explosion of multicellular life forms. Some of them are easily identifiable, like these cnidaria, but some other aren't. For example, this one, his name is Tribrachidium, and we're not sure exactly if it's an Echinodermata or a completely different taxon that is not known from other periods of time. Okay, so during the Ediacaria, we aren't at the Phanerozoic right now, but we are starting to increase the number of known taxa in the fossil record. And then nearest from the boundary with the Phanerozoic, we are able to find small Shelley fossils, which are very important for the fossil records because it's the first time we have biomineralization of small shells. So because of the taphonomic processes, it's going to be way more easy to find fossils who have hard parts like shells. So this is part of the revolution that is going to appear. And then we make a jump of a few million years, and that's it. We are at the beginning of the Phanerozoic. Here we are specifically in the Burgess shell, where you can see trilobites that can be easily identified here, or other like Anomalocaris, which is probably one of the first predator that had been found in the fossil record. So at the beginning of the Phanerozoic, we have an explosion of life forms, you can see here, and lots of very large taxa can be identified, like sponges, annelids, or mollusks. So this is why I chose to study the Phanerozoic, because I wanted to interest myself in the largest possible timescale to study biodiversity dynamics. And before the Phanerozoic, we don't have enough data sets to produce big analyses. So we are just here in Earth clock at the beginning of the Phanerozoic. And now we are going to have a look at the Phanerozoic itself and its biodiversity dynamic. You can see here that the Phanerozoic is divided into three main periods: Paleozoic, Mesozoic, and Cenozoic. During the Phanerozoic, we are going to have a trend toward the increasing of the ecological space. Here we are at the Ediacarian, and this is an illustration of the ecological space. Here you have squares representing three variables, like tiering from pelagic to infaunal, the way you catch food from suspension to grazing and to predatory. And here it's if your species is motile or not. Here at the Ediacarian, you can see that a few squares are occupied by species in this ecological space. Then a few dozens of million years after, we are starting to be in the Phanerozoic. Here you can see an increasing in the occupancy of the ecological space. And this is now the ecological space with, for example, with bivalves, which are suspension feeder. They are shallow infaunal, they are inside the sediment, and they are unattached, but not really, not completely motile. And you can see here a Phanerozoic trend toward the increasing of filling of this ecological space. Okay, so the Phanerozoic is divided into three main periods that are separated by mass extinction events. And these mass extinction events are going to be very, very important for the next part of the speech, because mass extinction obviously have a very strong impact on diversity dynamics. This is an illustration of Paleozoic. So where you can find obviously the vertebrates, especially fishes. You can have tetrapoda, but also plants on the land, trilobites, brachiopods, bryozoan, gastropods. And then right here, we have probably the most important mass extinction event, which is called the Permo-Triassic mass extinction, where approximately 81% of species disappeared, mainly because of Siberian traps. The traps in the fossil records are generally found as supervolcanism events. So here in gray, you have the extent of the area where lava from Siberian traps, like here, have been found. So these events are really gigantic. And you can guess that the release of carbon dioxide in the atmosphere has led to a dramatic mass extinction. So right after this mass extinction, we lost 81% of the species, and then we had new origination, and this is why in the Mesozoic we have a completely different picture of life, with mainly ammonites, gastropods, marine reptiles, or in the plankton you have the appearance of diatoms, for example. Okay, so we are at the Mesozoic, and then another mass extinction, Cretaceous Paleogene, which is perhaps the most known in popular culture, with disappearance of many species of dinosaurs, but also mammals. Okay, so this is one of the most famous curves of diversity dynamic through the Phanerozoic. So it is a curve built by Sepkoski in 1978, using a very large compilation of marine taxa, not species nor genera, but here the first is about families, which was most easy to number, from Phanerozoic here. And you can see several interesting trends. So you start with an increasing diversity during the Cambrian, which accelerates during the Ordovician, then a plateau with some extinction events here and here. Then the most obvious mass extinction is the Permo-Trias I was explaining right before. And then you have a continuous trend towards increasing of diversity during the Mesozoic and Cenozoic, with the Cretaceous Paleogene boundary and mass extinction here. Okay, so this is another illustration, because these diversity curves show the standing diversity, the number of taxa at a specific time, but it does not explain if it's, when you have a plateau, if it's always the same species. So you can have high turnover, but diversity which remains constant. So you can see here the fauna, which becomes extinct at each extinction event and renewed by new species. Sepkoski was one of the first to study this kind of curves using large amount of data, and he aimed to study carrying capacity. So is there hard limits to diversity on Earth due to competition? For example, if you have too much species at a certain location, then you are going to have a strong competition, and then to increase the extinction rate and to decrease diversity. So he tried to fit a logistic curve with a first increasing and then a plateau to his curve, which didn't fit because of the mass extinction, but several logistic curves during one for the Paleozoic and one for the Mesozoic, for example, seems to work. Except for the end here, because of a very serious issue with this curve, which is the pool of the recent. The pool of the recent is a taxonomical bias. It states that it's more easy to find fossils when you are near from the present, because fossils are more well preserved. So this increasing from Trias to the recent might be a taxonomical bias. You have also strong sampling heterogeneity, because some periods are well known with lots of fieldworks, lots of sedimentary rocks which held fossils, but some other are not as sampled as the other because there is not enough sedimentary rocks. Okay, so Sepkoski was one of Sepkoski's curves was one of the most studied curve of biodiversity dynamics, but it has many flaws. So in 2008, Alroy tried to use sampling standardization methods using new databases on genera and not families. And using sampling standardization methods, he found that the curve of marine metazoans, so I didn't tell you, but this is only marine and metazoans, so multicellular life forms. So Alroy tried to do the same, but while taking into account sampling bias, and he found a completely different curve. If he found again the Permo-Trias mass extinction, the Cretaceous Paleogene, but there is no plateau here at the end of the Paleozoic, and the effect of the pool of the recent has been removed. So we have an increase, but which is way less strong than the first. Alroy was also interested in carrying capacity, so he tried also to fit logistic and exponential curves to this curve of diversity, standing diversity through the Phanerozoic, and he found that the logistic curves were really good to explain this curve, and the exponential one does not. So he concluded by saying that marine life has reached carrying capacity, and then no exponential growth unlimited exponential growth could be found by studying this curve. So he proposed a mechanism of diversity dependence for this logistic growth pattern, which is supported also by a correlation test he made between this curve and a curve of origination and extinction rate. So we have standing diversity. The origination rate it reflects the proportion of taxa that appears at specific time beams, and the extinction rate states the proportion of species that disappeared at a specific time beam. So, for example, when you have a mass extinction event, of course, you are supposed to have a peak in the extinction rate. So there is a correlation between origination at a certain time and diversity in a subsequent time beam, which seems quite logical, because if you have high origination rate, so then you are going to create more species, and then the diversity level will rise. But more interesting, you have a correlation also between diversity and subsequent extinction rates, which is another proof of diversity dependence, because again, this shows that high level of diversity are associated with subsequent high level of extinction, probably because of the effect of competition from limited resources. A year after, Benton proposed a theoretical model without doing analysis, and he proposed that biotic factors, so such as competition, dispersal, genetic drift, were small-scale drivers that works at populational scale, but that large-scale drivers are mainly abiotic factors, such as nutrient input, temperature, catastrophic events like meteorites. In general, in the literature of the 2000, between 2000 and 2010 and before, generally geologists say that the main drivers of biodiversity dynamic are abiotic ones, while biologists and ecologists say that no, the main drivers are the biotic ones at large scale. So biotic and abiotic factors are intricated, and this is a last example for the historical background from Cardenas and Harris, which tried in 2010 to make some correlation between the origination rate and the nutrient input. The proxy for nutrient input here also for marine methazones was the isotopes of strontium and sulfur. And he found that indeed there is a correlation between nutrient input in oceans and the origination rate of marine life. So that seems quite logical. There is, if you have more nutrient input, then you will have more food, more abundance, and then more species that are going to arise. But Cardenas, Harris, Alroy, they all tried very coarse-grained techniques like correlation tests, which have many flaws. So now I'm going to present you my first results, which aim to disentangle the biotic and the abiotic factors as drivers of biodiversity dynamics on several taxonomic groups from throughout the Phanerozoïc.
Valentin, sorry to interrupt you, but Tonda has a question.
Yeah.
Hi, Valentin. I really enjoyed the presentation. It's a very nice summary of the geology and the history of life on Earth. Before we get to the results, I was wondering, what is your position on these two different views? The equilibrial view where the diversity is stable and the one where diversity increases exponentially. Like, what would you say is the more meaningful?
I think actually nobody is thinking that there is no limits of the diversity on Earth, like Benton, but they are more saying that there is exponential growth because the carrying capacity is far too high, and then diversity never reached it. But basically, it is a question I'm trying to answer with this analysis. But yes, the main aim was to find new evidence for carrying capacity and for limits to diversity.
Do you think it's possible that there is some kind of a middle ground, that it's not an equilibrium in the sense that there is a stable number of species, or it's not really exponential growth, but it's some kind of a growth without a clear limit, without a clear ceiling? Like, is there some third middle ground between these two extremes?
Yeah, but I think with the equilibrium theory of biodiversity dynamics, it's probably more something like that, where carrying capacity is an attractor and not a hard limit. So diversity fluctuates and is attracted to the diversity, to a specific diversity point. So with this model, you can have carrying capacity and never see any exponential or logistic growth.
Thanks a lot. I will maybe have more questions later, and maybe we can talk one-on-one.
Okay.
Thank you. Just one question. Tomáš Gavenčák had also one question, or not?
Yeah, but I'm also happy to ask later.
Okay, then
maybe we can ask very quickly, and if there will be longer discussion, we can postpone it. So you are measuring either taxa or number of genera. It seems like that both like on whichever level of granulation you choose to do this, it doesn't capture the whole picture, right? You can still have one group which you consider one group, but it actually has many subgroups, or you are looking at the subgroups, and then you can be missing some other group which is rich in a different way. I'm sorry, I'm not really a biologist, so I'm not really maybe using the right terms here.
Yeah.
So are there some more considerations or more metrics to use for this diversity, or is it just like those two, or is it one of them actually the right one? And again, that might be a longer discussion. I'm just curious for the results.
Okay, perhaps we will pursue at the end. I will just say one thing. There is a dichotomy in the publication on this topic between people who are trying to understand diversity dynamics using phylogenies, evolutionary trees, and those like Alroy and Sapkowski, and I'm going to do the same. We want to try to understand diversity dynamics at the largest possible scale, and at this Phanerozoic scale, the phylogeny is not really good. Then we are only using databases with the occurrence of taxa. Okay.
Thanks. Yeah, I would like to come back to that later, but
okay.
That's fine for now. Thanks.
Okay, so now I'm going to explain you my protocol on how I try to answer the question of the drivers of biodiversity dynamics in the oceans. So I will use taxonomic data sets, the largest possible. And what we need is a time series of diversity of origination rate, of extinction rate, and of energy surrogates as temperature and nutrient input. The first source of data was the paleobiology database, which is the most important fossil database, which can be completed by anyone who find a publication or fossils on the field. And something that is really important is that the PBDB is an occurrence database. So when you have a species, for example, this is a bivalve named Sauvagesia sanfilippoi, in the Excel sheet, you are going to have a line with the time interval, the country, here Afghanistan, Dhaka, and the name of the species. But this is one occurrence, but you can also have another occurrence of the same species at another line. It's not a database listing only the species and their ages. This is very important because the fact that we have occurrence databases allow us to use sampling standardization methods like in the Alroy fashion. The second database with the Neptune database, where we have data of microfossils were in cores that have been drilled during the campaigns of the oceanic drilling program. Here, each dot represents a drill. So there is a huge data for microfossils, which are plankton, basically. So, okay, we are also at the taxonomic level of the genus. I chose the genus because we have insufficient data for families, families which are also somewhat arbitrary with lots of paraphyletic taxa. But at the species level, we have also a problem with the taxonomy. For example, the delimitation of species are difficult in the fossil record. So the trade-off was here to take the genus as a taxonomic level. Okay, so I chose 9 taxa to study. These taxa, I choose them because they are well represented in the fossil record and well sampled. For example, we have scleractinian corals. We have brachiopods, which look like bivalves, but are not related to. We have also bivalves, like the mussels, gastropods, like snails. So these 4 taxa are well represented in the fossil record because they are shells, basically, or calcitic skeleton. And then we have also the marine metazoan to be able to compare the results with those of Alroy. So marine multicellular life forms, so corals, brachiopods, bivalves, gastropods, but also vertebrates and other arthropods like triglobites. And then for the Neptune database, we have radiolarian, foraminifera, Cocolitopharids, what the main constituents of cliffs of northern France, for example. These cliffs are made of tiny Cocolitopharids and diatoms. Okay, so we have the data sets. Now we need to build diversity curves. The diversity curves I computed using a technique called shareholder quorum subsampling, which is a coverage-based methods. The SQS tells you how many species you would find given a fixed coverage. The coverage is basically the proportion of singleton in your assemblage. If you are comparing two time bins, in one time bin you have no singleton. So singleton are taxa which have been discovered only once, so only one occurrence on the datasets. So you have a time bin with no singleton and a time bin with tons of singleton, which are basically rare taxa. You can suppose that there is a problem of subsampling, because even in a poor environment, you are supposed to have a specific proportion of rare taxa, thanks to species abundance distribution. So the SQS method is going to rescale the diversity of each time bin given a specific coverage, so a specific proportion of singleton taxa. So we have a measure of diversity, and then we correct this diversity using a sampling probability named the free timer that aims to suppress the pool of the recent. So the rationale is the following. Here you have temporal scale with 3 time bins. Here you have an example of a species which is present in the 3 time bins, and a species here which is present in the first, in the third, but not in the second. This is a ghost range because it's the same species, so it must be here. So if it's not here, it's because we didn't sample enough. So part timers reflect the sampling completeness of the fossil record. So by calculating the sampling probability, which is 3 timers divided by 3 timers plus part timers, we are able to rescale the diversity and to suppress the pool of the recent, which is mainly affected by ghost ranges. And for assessing origination and extinction rate, we are going to use the second-forward method. I will not go through the detail. I just want to add something for origination rate. I'm specifically talking about origination and not speciation, because we don't have phylogenies. So the origination rate is calculated with the first appearance of the species and not on reconstructed ages of speciation of taxa. Okay, so we have diversity, origination, extinction. Now we need energy surrogates. The first one is temperature. Thanks to a nice review of Scotty's in 2021, this year, we have a nice study of temperature through the Phanerozoïc, which is based on oxygen isotopes and on climatic belts. These are the climatic belts today, with the repartition of temperature through latitude. And these are the reconstructed climatic belt using geological evidence. For example, evaporites are rocks that form only in arid condition. So when you find evaporites, then you can track the climatic belts, and you are able to reconstruct them. So this is a curve of temperature through time during the Phanerozoic, and this is a curve of temperature based on climatic belts. also through the Phanerozoic, and then it can be compiled, and it has been compiled by Scotties to have two evidences for the rise and fall of temperature through the Phanerozoic.
Honza Smička raised the hand, so please ask the question.
Yeah.
Hello. Hi, Valentin. Greetings from field work. We're actually connecting from Statistical National Park. We're showing the reconstruction of biomes. Do you actually think that this kind of reconstructions exist, or are you aware of some other studies that would be doing this kind of reconstructions for like earlier periods of time, like from the tertiary?
You are talking about climatic belts?
Yes, yes. On this study based on like sediment types.
Yeah, you have the whole Phanerozoic, so 500 million years. So the tertiary also, but okay. It's at the resolution of the stage. So each points correspond to a mean of approximately 6 million years.
Yeah, that's actually super cool because we were looking for something like that with David some time ago and we couldn't find. So that's maybe something to talk about later. Thanks a lot.
Yeah. Okay.
And Tonda, Mahaj?
I was also wondering about the statistical corrections. You mentioned that there is one way to do it. Is it that there is one way which is the best way, or are there many different alternatives? Would other statistical corrections give you different results? Did you consider other options?
To correct diversity?
Mm-hmm.
There are other ways of correct diversity. Actually, another way is by using Bayesian methods on diversity estimates. It's with a Python package named Pyrate. And I tried to use this kind of Bayesian reconstruction, but then I have only a technical problem, is that these methods are very recent and they are not very well adapted to large scale diversity time series. They are mainly used for analysis through few million years and not more. So we have problems to compute the curves, and then generally they are almost completely flat.
Yeah, I can see that there are some technical issues. My question is practically, like, is there one best way to do this, ideal way, which is probably the one that you did? Or is it that there are multiple alternatives? We can't really decide which one is the best, and maybe we should use all of them and compare the results.
The problem is that there are not a lot of methods. Bayesian, this one with the free timers and the sampling probability, this is a family of methods. All have been built by Alroy. So there are 4 or 5 indices of diversity. I tried all these methods to compute diversity in this family, and they all give approximately the same. And then you have people who say that no, the raw data is better and we shouldn't correct the taxonomic data sets because it becomes just worse. But in this case, I'm seeing overestimation in each case, a strong put of the recent near to the present.
Thanks, that's really nice. Thank you.
Okay, so after temperature, we can also use the carbon isotopic ratio, which is a function of primary productivity, because with the rise of plants on Earth, plants can sort the carbon by their isotopes. They prefer to capture some isotopes of the carbon than others, and then when they are buried, when they are transformed to coal and capture, they are going to trap these isotopes to modify the carbon isotopic ratio, and then to modify the atmospheric and the oceanic ratio. But we need to be very careful with this kind of measure, because it's function of primary productivity, but also of carbon burial, preservation of organic matter, type of vegetation, because between C3, C4, and CAM plants, they don't have the same exact behavior with carbon isotopic ratio. We have also problems if we had a runoff of minerals and of sedimentary rocks that going to release these carbon isotopes through the oceans. So we have an effect also of tectonics and volcanism. And then we have strontium and sulfur isotopic ratio. Strontium isotopes are captured in the lithosphere, and when they are released in the ocean by continental runoff, then we can think that it's associated with a nutrient input. And the same with the sulfur isotopic ratio, nutrient input from recycling of organic materials in ocean sediments, generally associated with bacterial activity. But we need to be very careful with this free isotopic ratio, because they all are influenced by other stuff than nutrient input or primary productivity. And then for the method I'm going to use, I need to detrend this diversity rates and the energy surrogate time series. So this is, for example, the rise of the diversity of marine bivalves through the Phanerozoïc. You can see that there is a rise, which is not associated to a pool of the recent. So we need to detrend here to normalize and to analyze this time series. Okay, so now the aim is to assess causality between pairs of variables, which are our time series. And these causality assessments are generally done using correlation tests. Of course, there are tons of problems with correlation. One of the most important problems is that points must be evenly spaced in time. But I'm using for the paleobiology database, I'm using the geological time scale. And with the geological time scale, we have some stages who are extended of 1 million years, and some others that with the duration of 10 or 12 million years. So we need a method to assess causality more efficiently than correlation. So I used a method that begins to be used in paleontological time series analysis, which has been proposed by Sugihara in 2012, which is a convergent cross mapping and use dynamical systems. Convergent cross mapping can be used with any kind of time series. It can be neuronal activity or diversity, it doesn't matter, because it's a model-free method that detects nonlinear effects. So you don't need evenly spaced time points, only bin averaging, which is done in my datasets. And a very interesting thing is that it allows detection of unidirectional or bidirectional causality. Okay, so I will stay at the surface of things for convergent coarse mapping. This is time. At the left, you have a time series, and this time series can be understood as observation of a dynamic system. More precisely, time series can be viewed as projection of the behavior of the dynamical system. And thanks to the Taken's theorem, we can reconstruct a dynamical system using only one time series, but if each dimension of the dynamical system corresponds to a time lag. So this dimension corresponds to the time series, and this one corresponds to the time series with a lag of t, etc., etc. So for each time series, we can reconstruct a dynamical system. Then the causality inference is made by, you want to compare x and y and to assess if there is causality between them. So you are going to reconstruct a dynamical system of x and a dynamical system of y. And there we are going to embed these two variables into a third dynamical system. And by comparison of one variable with the other, we are going to, we can predict the position of a variable. More specifically, if there is bidirectional causality between x and y, when I'm going to use x here to try to identify the position of y, if there is a good relationship, if x can help to predict the position of y on the other dynamical model. In case of unidirectional causality, if x causes y, but y does not cause x, we would only expect cross-mapping to be successful in one direction. It's counterintuitive, but if a causal variable x leaves a signature on the affected variable y, then it is possible to map from y to x. So with y, we are there is going to be some success to predict the position of x, because as x causes y, there is information of x in y. It's probably not completely clear, but perhaps these figures might help a little. At the left, you have an example of bidirectional causality between x and y. The manifold represents the dynamical system of x. Here you have the dynamical system of y, and you can see that here we are successful in predicting the position of y using x, and to predict the position of x using y. So in this case, we have a bidirectional causality, so that x causes y and y causes x. When you have asymmetric causality, where x causes y, but y doesn't cause x, then you will not be successful in predicting the position of y using x, but the reverse will be true. And there is two ways to assess significance using convergence cross-mapping. With the convergence of the results, when you increase the library size, which is the sample, the number of sample of time series, And you can use also surrogate time series, for example, a random time series that are compared with the actual results. And these are the results. So I try to represent the networks of causalities for all the taxa. So for example, here we have the bivalves. And in the bivalves are represented the origination rate, diversity, extinction rate, and then the energy surrogate, temperature, carbon, strontium, and sulfur. When there is an arrow from temperature to diversity, it means that a causality has been detected from temperature to diversity. If you have two arrows like between diversity and extinction, it means that there is a bidirectional causality. Okay, so here we have the bivalves. Here we have the gasteropods, the brachiopods, the corals, the whole marine multicellular life forms. And then here we have the diatoms, the foraminifera, the Cocolitopharids, and the radiolaria. You can see that more significant arrows are present using the Neptune datasets. It's quite logical because I said earlier that with the paleobiology database, the time beams are the stages of the geological timescales with a mean of 6 million years. But with the Neptune database, we have absolute value. So the average, I averaged the beams, which are extent of 1 million years. So as a result, the time series using the microfossil data are longer than those with the paleobiology database. And if these networks seems to be chaotic, there are some interesting patterns. And when you try to look at what are the most strength links, you can see that it's mainly between biotic factors, origination, extinction, and diversity. Generally, between these three variables, we yes, Arnost?
Arnost, we don't hear you. Still
no.
Not. So maybe later.
Okay, so it seems that there is a strong signal between biotic variables, and that taken globally, the networks show weaker relationship between the energy surrogates and the biotic factors. So a first conclusion of this CCM analysis is that...
Hello.
Yeah.
Can you hear me? Sorry, I ask you later. Can you hear me, please?
Yes.
Yeah. Sorry. So quickly, I finally found how you recognize causality in your system, but it is the simple idea that you plot the axis so to get a function, isn't it? And if the function is strictly increasing or strictly decreasing, you are talking about bidirectional causality. And if it's a function, you are talking about causality. But it's true for system without memory. It was the lesson on the faculty that they asked us about this problem and then showed us the hysteresis curves. So if the system has memory, it doesn't work. So you can't, and the system is a memory, isn't it?
With the CCM method, you can specifically try to analyze two time series with different lags. So you are able to identify if there is a memory between the time series. If you can, and I'm going to show the results thereafter for some variables, we are able to show that there is a memory of approximately 6 million years, because when we look at the strength of the causality, so which is of course the ability to predict one variable using one another, then it's going to shade after 6 time lags.
So you are saying that the system controls for the memory.
System controls.
System is controlled. That memory is controlled in the method.
Yeah.
Oh, nothing. Yeah. So sorry for interrupting you. It's
no,
just
very
okay, important.
And, yeah.
Okay. Thank you.
Honza Smyčka wants to add something.
Actually, Arnošta, I think the memory the memory Valentin is talking about is mostly given by like the structure and scaling of the data. But like if you think about the mechanisms of like feedbacks between like origination, extinction, and diversity, I mean, I would like without any other evidence assume that like that there is no memory and that this is like really a Markovian process, that like the diversity reacts to the actual value of extinction and rate and of origination rate, and the extinction rate reacts to the actual value of diversity, and not looking like multiple million years back. Or why would it happen, like this like longer term memory, or like
I'm not sure if delay is memory, but you are saying that it's system without memory because memory is too short, isn't that's what you are saying.
What is the difference for you?
It was actually why I asked the question, because I tried to discuss this point, whether the memory can affect. I'm not a paleontologist, so I'm not sure if the memory can be so short or it's longer.
What is for you the difference between delay and memory exactly?
Memory is that system is determined by some previous states, and it the delay just react to the state of the same parameter. The delay is memory between the same parameters, but the system may depend on some other parameters you aren't modeled, you don't have in your data. So
okay.
I think it's an interesting like discussion maybe for the coolers, but I think that like normally in evolutionary models, like a lot of people assume that evolution is Markovian, and it's not something like specific that like Valentin would come with. I think like all the models that like Valentin uses, I use, I think Tonda uses, I mean they are basically all Markovian, so they are only dependent on like the last previous state.
Okay, that I never heard that one should say that that one can learn causality from analyzing, even if we suppose Markovian process.
Yeah, okay, this is the point.
We can guess that there is a causality of the direction, but this system pretends that we can learn exact direction of causality from it.
Yeah, I see the point.
Okay. I think. Thank you for the discussion. Thank you.
And Tonda?
I had a related question about the methods, because the methods are very like appealing, very sophisticated. And I was wondering if you can maybe like take it back a little bit and like explain in very basic terms how the methods work, right? Because I guess like you have the time series for temperature, diversity, and do you correlate these time series, or how do you identify the causality? I feel that was one of the like issues that we have been also discussing now. Could you like in very basic terms explain the principle?
It's basically because the correlation, of course, doesn't mean the causality. So some people try to use more refined methods to identify more precisely causality. And I think almost all methods that says that they are handling causality, they're all trying to predict. So if generally it's when you can predict y using x, then you can assume that there is causality from x to y. This is Granger causality, I think. Granger causality is about the ability to predict one variable using one another. And in this specific method it was a counterintuitive point. It's that they assume that it works in the reverse. So in the causality in the sense of Sugihara, if X causes Y, then there must be information in Y that is information from X. So if X causes Y, then you must be able to predict X by using Y. But all of these methods are trying to predict time series using all the time series.
I see. So you use one time series and you try to predict the other one, then you switch it and do it the other way around. And if you get better results, that's the direction of causality.
That's right. Yeah.
Thank you.
Okay, so we can continue, I think.
Because yeah, you see that these little schemes are representing the ability to predict. Here you have the location of 3 location points of X, and through a dynamical system, we are able to predict the position of Y on its own dynamical system. And in this case, it's not the case. Okay, so we are here, and there seems to be mainly an effect of biotic variables between them. So it seems that for most of the taxonomic datasets, except gastropod perhaps, which shows a strong signal from carbon to origination rate. All the diversity dynamics of the main taxonomic datasets seems to be driven by biotic factors such as competition, dispersal, genetic drift, and so on. But we are going to going through a specific example. Let's try the example of Cocolitopharids. So we have unicellular taxa. Here are shown the diversity curve in black before the trending, the green origination rates, and in red extinction rate. Here you can find the Cretaceous Paleogene mass extinction, which is associated with a peak in origination rate. This pattern is is very common in all time series. And it is known from perhaps 30 years, when you have a mass extinction event. Right after, you have a peak in origination, and the main hypothesis is that you have lots of space. So the survivors are going to disperse into empty niches, unspeciate. So extinction peaks are associated with subsequent origination peaks. But these are only correlation. And here you have temperature, carbon, strontium, and sulfur. So this scheme represents convergent cross-mapping analysis, but windowed. So this square, for example, represents, so I don't see exactly. Okay, so here we are trying to assess the causality from carbon to extinction rate. So the effect of abiotic factors on extinction rate. So for example, this little square represents the conversion cross-mapping analysis of 9 data points. So this locality from carbon to extinction rate, and the strength of the color represents the strength of the causality. And so here we are studying the 9 points here the 9 points here. So this is a sliding window that shows you the local convergent cross-mapping throughout the Cretaceous, Paleogene, and Neogene. And then here we are increasing the size of the window. And when you are here, it's the global convergent cross-mapping analysis. And here you have a convergent analysis of approximately, I think, 30 data points. Okay, so how to interpret these results? When you have a red color here, it means that there is local effects, and here it means that there is a global effect, a global causality. And you can see that the interesting point here is that there seems to be a local effect just right during the Cretaceous Paleogene of carbon to extinction rate. So during the Cretaceous Paleogene crisis, there are 2 main events: the fall of a meteorite in the Yucatan in Central America, and the Deccan traps. So same fashion as Siberian traps, lots of the massive release of carbon dioxide in the atmosphere, and the meteorites provoke the release of particles in the atmosphere that probably inhibits photosynthesis. So there is a global breakdown of ecosystems during this period, and it must have affected the carbon isotopic ratio. It's very significant in any time series of carbon isotopic ratios. So here we are able to find that there is a causality between from an abiotic factor, carbon, towards the extinction rates of the Cocolitopharids. You can see here the same with the temperature. It seems to be a little more global, but it's basically centered on the same points. This is with strontium. You can find here the same pattern, and with sulfur. So the four time series give very congruent results. There seems to be a strong effect of all abiotic variables towards the extinction rate. But this effect seems to be local. So now, I'm sorry, I don't see, I'm not able to see the title. When the interesting thing is that there is an effect on extinction rate, but it seems to be another, the same effect with origination rate. And then when you are looking at the causality between biotic variables, like here, the effect of diversity on extinction rate, so diversity dependence of extinction rate, then you see that the signal is mostly global. There seems to be a little stuff here locally, but the effect seems to guide the extinction rate throughout the whole studied period. So there seems to be an effect of diversity to the extinction rate. And when you are looking at the diversity dependence of origination rate, it's exactly the same, and it's even stronger in most cases. So this is very interesting because the carrying capacity seems to be supported by our results in Cocolitopharids with the diversity dependence of extinction rate, but also diversity dependence of origination rates. And so the attractor of diversity of the carrying capacity is here, and that this carrying capacity is probably locally modulated by catastrophic abiotic events, but not by temperature, for example, does not seem to modulate the diversity dependence of extinction on origination rates, except during these very specific catastrophic periods. And last, this is origination to diversity causality. Then you can see something interesting. The diversity seems to be mostly driven by origination before the Cretaceous Paleogene boundary. So if origination rate is the main driver of diversity, perhaps it could be this pattern that we are below the carrying capacity during the whole Cretaceous. However, the effect of extinction rate on diversity does not seem to show a significant pattern. We only have a global effect from the extinction to the diversity, which is coherent. Now,
Valentin,
yeah.
Do you have some comments in chat? Some general comments to the diagrams. Arnošt and Tonda posed them. Could you, Arnošt, maybe elaborate it more as a question? Or
It isn't important. Sorry,
okay,
it was about the impact of the meteorite. It was physics, not paleontology.
Okay.
Sorry.
Then we can continue.
Okay.
Sorry.
It was the only question. And now let's take the example of marine Methazones. Again, with diversity in black, origination in green, and extinction in red. Here we can find the main mass extinction event, the Permo-Trias, with a high peak of extinction rate, followed by a peak in origination rate. Then when we do windowed convergent cross-mapping analysis, we are also able to find diversity dependence of extinction rates, so probably mostly driven by competition. We are able to find diversity dependence of origination also, probably driven, for example, by dispersal. If you have more dispersal, then you are more susceptible, you have more probability to have a sympatric or allopatric speciation. But there is also a modulation of the carrying capacity by abiotic factors, which is only local. So there is the results are mostly congruent, except for for two variables, for two causality links, which seems to be very idiosyncratic and specific to each taxa. And this link is from the rates to the diversity. And here we are able to find that before the Permo-Trias mass extinction, the diversity is mostly driven by origination, and after it seems to be mostly driven by extinction. So these are my last results, so I am not sure how to interpret them, but during the Permo-Trias mass extinction is really a complete breakdown of taxonomy and of ecological space. There is a complete rearrangement of the species and their relationships. So perhaps it's linked to variation between ecological types before and after the Permo-Trias boundary. Here is a suggestion. For example, you have the proportion of motile animals during the Phanerozoïc, and you can see here the breakdown of the Permo-Trias. Thereafter, we have way more higher proportion of motile animals than before. It can be with also the proportion of buffered taxa. So this is a study of Bambach in 2002, and this shows the proportion of taxa which are sensitive to variation in carbon dioxide, and you can see that right after the Permo-Trias, you have a selection of species that are less sensitive to environmental variation. It might be another explanation. And then the convergent cross-mapping also allows to produce, to compare results of causality strength with time lags, so with delays, as we were talking right before. And this scheme represents you the mean of the causal links for the 4 microfossil datasets with the diversity to extinction, the diversity to origination, the extinction to diversity, and the origination to diversity. So this represents the overall causality strength between biotic factors with a delay, with a synchronous effect, with a delay of 1 million years, 2 million years, 3 million years, 4. And you can see here that there seems to be a global pattern with an effect, a remnant effect between biotic variables, which has a delay of between 4 and 6 million years. So my interpretation here was that perhaps when you have a disturbance in the system of biotic variables, then the system will be there will be a delay of approximately 6 million years. And I didn't make it with the abiotic variables because the patterns were not congruent between each other. And last thing, thanks to an idea of Hamza, was with the comparison of the diversity dependence of extinction and origination rates with and without a lag. So these squares represent the 9 data sets I analyzed. So it's the causality strength from 0 to 0.6 from diversity to extinction rate, with a synchronous effect and with an effect of 1 lag. And it seems to the effect of diversity to extinction rate seems to be mostly diachronic, because most taxa sets are have a less causal value with a synchronous diversity to extinction rate effect. However, it does not seem to be the case when you are looking at the effect of diversity to origination rate. Okay, so there is a lag when you are studying the causality from diversity to extinction rate, but there is no lag with diversity to origination rate. And the idea here was perhaps it could be a bias of ephemeral speciation. So the ephemeral speciation model tells you that of course, we miss lots of species, but when we have higher diversity, then we are going to miss higher proportion of species that speciate and immediately after become extinct. In this case of pattern, it will be very unlikely to find these ephemeral species in the fossil record. And so there is going to be a bias in the causality inference. For example, we have the origination rate will be affected. And if we miss some species, we are supposed the miss of species and of originations will not have a signature with a lag. It will be synchronous. So I tried another method to make things a little clearer. I used another method to assess Granger causality, transfer entropy. And the transfer entropy has an advantage. is that it can remove the effect of a third variable. So it's called conditional transfer entropy. And here I removed the effect of abiotic variable, because the ephemeral speciation model should affect only biotic variables. So we should see a stronger effect. And it's probably the case because here you have the strength of the diversity to extinction and diversity to origination. And when we remove the effect of abiotic variable, then except for brachiopods and corals, we have always a stronger effect of diversity towards origination rates than to extinction rates. And this could be caused by the ephemeral species, because here you have carrying capacity with the diversity dependence of turnover rates. And if you have, if you miss a certain proportion of ephemeral species, then the absolute number of species you are going to miss will be increasing with diversity. So you are going to identify less extinction events. So you are going to underestimate extinction rate, but you are also going to underestimate origination rate. And the consequence might be a decreasing effect of the diversity to extinction, but an increasing effect of diversity to origination. And this might be explained by this scheme. The ephemeral species bias the results with a synchronous effect on origination rate, and with it probably overestimates the effect of diversity on origination rate. So I'm going to conclude this presentation. The conclusion is that logistic models are good, but they are not sufficient to test for carrying capacity precisely. So I've done this using methods of causality assessments such as convergent cross mapping and transfer entropy. The biotic factors seems to be the main drivers of the diversity dynamics, with diversity dependence of extinction rates and diversity dependence of origination rates. The diversity can be driven by extinction or by origination. This seems to be very idiosyncratic and taxon specific. And abiotic factors seems to punctually, locally modulate the carrying capacity. And finally, the ephemeral speciation might bias the results, but the ephemeral speciation we have seen might bias the diversity dependence of extinction or origination rate, but we still are able to find diversity dependence of extinction rate, which seems a very nice evidence for carrying capacity. Thank you for your attention.
Okay, thank you very much. You can see clapping our hands, and I'm sure there will be also questions, either the already announced during your presentation or new ones. The floor is open for the discussion. So Tonda or Tomáš Gabenčák, we can back to your questions.
Yes, thanks for the nice presentation. There are several things, but let me start with the one I asked about earlier. Mm-hmm. So you mentioned several measures of diversity. There was the number of taxa in the beginning. There was this was the graph, which when over to 2000 currently. There was the graph of genera, which was in the hundreds. Then there was some diversity index. I'm sorry, I might be misremembering some details. Mm-hmm. And then you also use some corrected diversity, I believe.
Yeah.
And how much do you think this like what is the correct measure, or what is the why did you choose the measures that you do? Because it, for example, seemed that the graph of genera was actually very chaotic compared to the graph of taxa in the beginning. So what are the considerations for choosing the right metric here?
I think there are two main problems with the diversity curves in using the fossil data, which are the pool of the recent and the signal ellipse effect. The pool of the recent is probably the main cause of the big trend in Sepkoski curves. So it seems quite logical that removing strong bias like pool of the recent might produce more chaotic curves. And you have also the signal ellipse effect, which tells you that you always underestimate the date of appearance of the species, and you always overestimate the extinction event. So there is going to be a lag in the extinction on origination rate, because when you see for the first time species, it does not mean that it just speciate right now, but the speciation was before. And when the last time you see a species, the extinction is not just at this time, but right after. And the metrics I used try to handle these two problems of the pool of the recent and the signal effect. And yes, it seems to remove the main trends of the diversity curves, except mass extinction events.
Thanks. That answers one part of my question. The second part was what are the things that you are counting? So, for example, I can imagine if you have, and again, I'm not a biologist, so I might not be using the right terms here. But if I imagine a species that has a lot of subspecies, or like a family which has a lot of subspecies, then counting all of them equally to another group which only has very few subspecies seems unfair in a way that, I mean, if you have some kind of insect that has like hundreds of subspecies that, for example, fill a similar role, just like in slightly different niches, and sort of are very, very close, both genetically and phylogenetically. And you have other species which are actually quite far away from each other somehow. That seems that counting them in the same way makes sense in different contexts, or you could have different metrics. So the question is whether there is one right metric which is the right one that you are looking for, whatever the desiderata for that are, or if there are multiple metrics you could be looking at, and if there are differences.
I think your question is more about speciation concepts that are with metrics, because in paleontology and using fossil data, we don't have any data on population exchanges of genes, stuff like that. We are only using morphospecies. So basically, species in my data sets are groups of individuals that specialists are able to distinguish from other ones. So it's a completely practical and methodological approach for species. And this is why this concept of morphospecies is supposed to be somehow close to the biological concept of species with flux of genes between population. But okay, so species are defined as the smallest groups you are able to distinguish from others. Mm-hmm. So in this sense, in paleontology, it's very rare to have subspecies, because we define precisely species as the minimal level of distinguishable species.
Right. So my question was, let me just ask one more concretizing question. Okay, so you have species. Thanks for the definition of that. But I guess you could still, I would guess that the experts on recognizing those species will still have groups of those species, right? Like some families, or I'm not sure mm-hmm. how you call them properly. And it might be different if you count the recognizable species or like distinguishable species, mm-hmm. or if you group into families and just count the families, for example, because it can maybe at one point in history you have actually a lot of families that are like very distinct, mm-hmm. and at some point you have a lot of less families but more species which are distinct but maybe close together somehow. So if you would, do you think if you would use a different kind of coarseness of this metric, you would get different results, or has it been
you are saying, do I have different results if I'm doing the analysis at the scale of the species, of the genus, or of the family? Mm-hmm. There is going to be some congruence, but not perfectly. The main problem with this is that it's very difficult to work with linear categories, family, genus, species, because except the species who are defined as a minimal entity, families are very subjective. Because if you are interested in some morpho-anatomical feature, you are going to say, this is a family, this is another family, because I think that there are two very different groups thanks to this specific morpho-anatomical feature. But another specialist will say, no, there is no two families, there is 3 or 4. So the families are mainly a scientific consensus, and they mainly reflect it's mainly a language that help us to communicate between each other. So it seems I have, of course, different results when I try analysis on genus of families. So I chose to take genus because of my experience of paleontologist. It was a good trade-off, because genus, general species, it's quite delicate, because in the world of paleontologists, you have many people who find 2 fossils that are not in the same locality. They are slightly different, but we are not sure. So they produce two species. So it seems to be very difficult to reproduce the work of definition of species using fossil data. So the genus level seems to be not the most subjective, but the less subjective level of because it's based on a more general level of overall similarity to distinguish taxa.
Thanks. So if I would paraphrase, I would say I would say that you could look at different levels of the hierarchy, but the data there is not very I don't know, like stable or well defined. So so that the level you're looking at is one of the the one where the data is most based on hard facts, if I would if I can say so.
And yeah, but I tried something like that, but not by using families or genus. But for example, when I study Methazones on bivalves, then I am at different level of the phylogeny because bivalves are Methazones. So here you can find similarities and difference between groups and their subgroups, but still at the generic scale.
Mm-hmm. Thanks. Thanks for answering my question. I apologize that it was so long. I would let other people ask.
That's okay. We have other, so I think Jan, Honza Smička, and then Tonda.
I would just add to what Valentin was saying, because he was talking about the merit of like choosing the genus level for the analysis, but there is also like a practical view, or at least how I perceive it as a non-paleoecologist, that vast majority of these large-scale paleontological studies are done on genus level. So also if one wants to compare to the others, it's a good idea to pick the genus level. But of course, scaling of these things to let's say coarser categories might be interesting as well. Well, I don't know.
Yeah, the problem with families also is you increase the risk of having paraphyletic taxa, which have which have no meaning for analysis on diversity curves. So.
Just to clarify quickly, I wasn't just thinking about switching to a different level, but maybe I can imagine metrics that would somehow weight the families differently based on the number of species, or I guess there might be other basically weighted approaches, but not just going coarser. But yeah, that would be a very different discussion. Sorry for that, and thanks.
Okay, so Tonda and then Arnošt.
Yes, I was wondering about your results, right? Because you find that the origination extinction are linked to diversity, and they're more tightly interconnected than they are with temperature. I was wondering if this, maybe to some extent might follow from the logic of the question that actually the origination influences the number of species. When some species go extinct, the diversity goes down. So variables will be necessarily more interconnected as opposed to, for example, temperature, which is estimated independently. Do you think that this could influence the results?
Sorry, can you repeat?
That you found that speciation, extinction, diversity are more interconnected with each other. There's more relationships as opposed to environmental factors such as temperature.
Okay.
And I'm wondering if temperature was estimated separately, right? From some historical data. And these three variables, new species originates, there is more species as a result. Some species go extinct, there is less species. This is why, simply from the logic of the problem.
Okay. There is a link between species and, if you can remember, for the estimate of temperature, we have climatic belt and we have oxygen isotopes data. For the climatic belt, it's mainly constructed using rocks, not fossils. But for the oxygen isotopes, for example, if you are looking at the curve at the Paleozoic scales, we mainly used brachiopods. So you take a piece of a shell of a fossil brachiopod, and then you analyze it, and you have the isotopic ratio of oxygen. So it's linked in this way, but I don't think it might bias the results, because we are doing this measure have been done using lots of fossil specimens. The main bias we could have is that with the temperature or nutrients, I'm at a worldwide scale. So when I show you the curve of the temperature at a worldwide scale, it's risky, because there are lots of different situations in the Pacific, in the Atlantic, in the Tethys Oceans. So the merging all these time series might be risky and might blur a signal of abiotic factors.
Yes, so like there was actually a related question. Maybe we will have more time for the sources of biases and errors. But it seems like for this question, you see that all of these variables, temperature, speciation, extinction, diversity, like they are fair. It's a fair comparison. All of them have the equal chance of being supported, right? It's not that one is more precisely estimated than the others, and that's why you get the.
Yeah.
I see.
Except perhaps with the bias of ephemeral speciation, but it's not a problem of resolution, but of underestimation or overestimation. Yeah.
Arnošt?
Thank you, thank you, Valentin, for nice talk. I didn't. Honda Smichka makes me contribute. I didn't want to contribute to the discussion, but Honda supported you, Valentin, so I support Mr. Gavenček for example, in this discussion. You made the talk in the way that there was something about it, because I was thinking all the time about the same thing as Mr. Gavenček, and I think that it's important issue, because from my experience with data, was data on land snail shells. When you measure differences between morphology of land snails shells. On the north, there are bigger differences between individual species than in the south. So if you recognize the species just on morphology, or genera just on morphology, you probably record less or the same number of species in the south than in the north. Even though opposite is true, just because the difference between morphology is smaller at the south than in the north. And if we replace time for space, which is a popular thing for paleontology, I can imagine then in some period there are more climatic or another variables as in the south, and in the some periods there are more climatic parameters as in the north. So maybe the resolution of species and the species or general level just from morphology may vary just because of ecological morphospace.
Yeah, it's very difficult because we only have morphology for fossil data, obviously. So the only thing we can do, because there is a problem with the taxonomic databases such as the PBDB, is that anyone can fill the database and add species. There are a lot of taxonomic bias. For example, on my group of interest, the Redist, some species have been described 300 years ago. Some are described now. This is not unified. So for certain groups, like scleractinian corals, the entire database has been carefully revised by specialists of scleractinian corals. I think, I'm not sure if we can do anything better than to ask specialists of the groups to specifically evaluate this kind of issues which are related to species delimitations. So I only add this revision for corals, but of course for gastropods, bivalves, or methazoans, it's way too huge to try correct these kind of issues that we do not correct, obviously, with the help of specialists, but
yeah. I didn't want to compromise the specialist, but I may imagine some control mechanisms, like now paleontologists control for sampling effect, which is famous RAUP and so on. So there might be some calculation, some computation machine which would control for this effect, for the
like
effect of
weighting differentially species with their locality.
Yeah, that in tertiary, whereas tropical climate, the number of species is actually larger than recorded from morphospace because individual species are closer, morphologically closer to each other. But I am just was thinking of this way, so I were like that Tomáš Gavenčík, Mr. Gavenčík, raised this question.
Mm-hmm.
And so time for the last two questions. So I'm not sure if Honza Smyčka wanted to ask, but definitely Tonda.
Maybe just like a quick question. You said that you want to find evidence for carrying capacity. Did you find the evidence?
Yes. I found the effect of diversity on extinction, on origination rate. So yes, I don't try to fit logistic curves, but the very strong effect of diversity on extinction rate and origination rate seems to be present in 7 of the 9 taxonomic data sets. I have very strange results with the corals, which shows practically nothing. So that is very strange. And for diatoms, which have the peculiarity of being photosynthetic, but mainly yes. And an interesting finding is that there is the diversity dependence of extinction, which is the main driver of carrying capacity, as hypothesized in ecology and in paleontology, but also diversity dependence of origination rates.
It's very cool work.
Oh, and Honza, the last question.
Yes. Actually, I was just wondering, and I think we've been actually discussing about that. You say that you have like the weird results for the atoms and for the corals. Could you just comment on, well, because like one thing that like distinguishes corals from the rest of the data set is that they're mostly asexual. So maybe it might somehow interfere with the previous questions from Tomáš Gavenčík and from Arnoš, that the definition of species in corals might be different, and or definition of genus in corals might be different, and generally the mechanisms, how like the evolution is going on, might be different. And another thing is for diatoms, that diatoms are actually photosynthetic, or at least part of them.
Yeah.
Do you have an idea how this like does that? Do the differences in diatoms and in the corals from rest of the data set somehow like show that like these two like traits might be important, or is it like completely?
But it's not as simple, because the patterns in corals and in diatoms are completely different of the others. But it does not mean that the patterns in diatoms looks like the one in corals. And also in Cocolitopharids are mainly autotrophic also. Okay. So it does not work. So it seems to be more complicated.
Okay, thanks.
Okay, so I think our time is up. It's exactly 12 o'clock. So I would like to thank again Valentin for his very nice presentation, and
thank you.
And to all the participants for their discussion. So It was really nice time. And next Thursday, we will have Alice Stashkova, and we will switch into philosophy. So have a nice rest of the day and the next days.
Thank you.
Thank you. Thank you.
Thank you. Bye-bye.
Bye.
Bye. Thanks.
Bye.